reasonable enough to start. Yeah, and we will record this meeting, so it will go on the website, so people that missed it can see it. I'll host today and try and chair, so we can stick roughly to time. We have 3 talks that we're going to go through. If you want to go to the next slide, Nicolina. Basically, the theme today, if you've been at the other NanoHarmony webinars, is the phase 2 to 3, and I'll go into exactly what that means later if this is your first NanoHarmony rodeo. But it's really looking at the technical development, commenting and approval rounds, so the kind of bit that resides within the WNT of the OECD process for how test guidelines is arrived at. This is a series of webinars, and we've now at the bit where we're looking at how the long bit of work in the middle progresses and how we can get that to go as fast as possible. Next slide, please. We will have some Mentimeter bits. Nicolina will put the link and code in the chat for you, but these are it. And we'll get to some test questions later, like which country you're from, what do you do, just so we can get everybody into it. And then there will be a set of questions that we've had recurrently in each webinar, which lets us capture different parts of the feedback from everybody that's in the room, because we won't get 33 people to speak today. So that's why that's there. So please participate. You can either do it on a phone or just open a different page in a browser. Next slide, please. So NanoHarmony, the aims of the project is really to take the inputs that are from all the ongoing projects that are dealing with nanomaterials and possible standards, issues, measurement issues, etc. Work with the stakeholders, regulatory bodies, standardization bodies, and what available data and methods that are within all kinds of projects. Try and put that through the NanoHarmony project and work out how we can make this process of developing and validating test methods for the OECD as fast as possible. We will look at one of the phases today, and there will be a little more detail on what they comprise in the next slides. Really the key bit is today about the test guidelines and the guidance document that are needed to respond to regulatory needs when we have a new technology like nano. So there'll be things that we were assessing for standard chemicals, if you want, that you can't assess for nano, and then we need a new set of standards for that safety to be reported and assessed. So it's to support the uptake of new and complex methods by the relevant and sufficient guidance on testing. Try and also include how you interpret data and the reporting via templates, so that the whole process is as simple as it can be made, both for the industry that has to report, but also for the regulators that have to interpret. It's about harmonizing the testing methods so that we make sure they're robust and reliable for governments to base their decisions on. We're very mindful of trying to reduce animal welfare issues, to work towards the 3 R's principle of reducing testing. It's also really important that people developing test guidelines are mindful of the economic implications of testing. You know, it could be scientifically very elegant and elaborate, but then it's so expensive that no CRO is going to provide the test, invest in what the infrastructure that needs to be there to let the test run. If it's not run, then obviously nobody can avail themselves of them. So we have a very beautiful piece of work that isn't going to be particularly usable, even if it would have been useful. And it is about this all feeding into broad accessibility for customers to use it, or industry to use it. So the OECD member countries are the ones that have to agree by consensus, and consensus is important. We'll probably meet a few incidents today where that consensus actually comes into play, that one country being not in agreement can be enough to stop a final TG or guidance document going through. But we'll see what comes up in the speakers' slides later. Next slide, please. So within NanoHarmony, just really short, there is a task, a work package up front, which has specific test guidelines in it that are at various stages of development. They are human and ecotox effects measurements, the green ones. Then there's phys-chem measurements, the yellow ones as well. And it's really all about taking, in some cases, tests that were there before and making sure they can be run reliably and reproducibly for nanomaterials of various, I was going to say shapes and sizes, but that's a bit of a pun. Various forms of nanomaterials can be run through these tests, and also highlighting which forms just won't work for certain tests. Next slide, please. So this is where we were at the start of NanoHarmony. 3 of those arrows over to the left have now caught up with the arrows that are in the middle. This is the whole process from start to finish of what you need to go through for developing a test guideline or guidance document around a test guideline. It starts with obviously ideas at the front end of things that could be done better or standardized. You make a proposal to the WPMN, the Working Party for Manufacturing Nanomaterials. And once you've made that, you then hone that argument into an SPSF, which gets submitted and verified first at WNP, commented on there, and really finalized before it gets submitted to the WNT. Mar will go through this later, I'm sure, so I'll skip over it fairly quickly. Then there's various stages of commenting round and approval in there. And then you get to another orange box, which is the one where we really could gain time, because you can see this middle box is from 2 months to 1 or more years. So depending on how well prepared you are, this could go really quick, or it could take an enormous amount of time. We will see different experiences today, but none of them are what any of us would possibly initially have thought of as really quick. 2 months is certainly something I've not seen in any of the examples we worked on. And then it goes into standard rounds that are bound by meeting dates again. So parts we can't really accelerate. The commenting rounds, the kind of WNT expert group meetings, and the final submission and approval stages by WNT. We've tried to work with all these projects to get as much feedback between them. So people that have been through certain stages are feeding back to the ones that are coming earlier in the process to try and learn as much as we can. And these workshops, webinars that we're running are really focused on certain stages, which I think is in the next slide, please. So here you can see the phases we've identified. It really initially had phase 1, 2, and 3 in it. And during the NanoHarmony project, we've identified a phase 0 and a phase 4. Phase 4 was really spurred on by contact with industry, where they found that certain tests, while the guidelines were there, weren't that easy to use because contract labs weren't making them available. So therefore they couldn't order the tests to be done. Contract labs in some cases found them expensive to undertake and couldn't see a market value in running them, which obviously is something we then had to feed back into the phase 0 at the start, where the initial idea goes from being the scientific idea of whatever project is developing the piece of work to something that has still all the central parts of that idea, but is thought of in a way where it gets simplified to be kind of mass usable right from the start. And that becomes one of the key objectives. So the phase 1, from project proposal to SPSF, we've had that workshop, and today is really about phase 2 and phase 3. How do you prepare for the different deadlines and targets within this process? and how can we make it go as fast as possible? Where are the holdups that people have seen, and how they dealt with them? And that's where I'm hoping everybody else will feed in from their personal experiences as well. We've got 2 case studies we're going to work through, but please feel free to add in. We'll take a couple of specific questions between each presentation, but hope to have a good debate at the end. Next slide, please. So the aim of today is to identify and understand what those challenges and barriers are to the development process bit we're looking at in stage 3 and 4, or phase 3 and 4. Get feedback from anybody in terms of your experiences of developing, using test guidelines, which bits should have been ironed out in phase 3, 4 during the technical development that became a problem later once the test guideline was accepted, for instance. But also really look at how we take all this knowledge that we're building and put it into a legacy that lasts beyond NanoHarmony, in terms of how do we establish and share the best practice for the future. One of the things that have come out is that people rarely do this more than once. There are few people glutton for punishment like Frank, who will go through these things more than once. And we all benefit from it. But most people arrive at this completely fresh and really could do with some very simple guidance on how do we get into the process to make it run as effectively, efficiently, and smoothly as it possibly could. And sometimes that's just about preparing in time for something that maybe not coming till a year or 2 down the line. But certainly if you don't prepare for it, you will find that delay later on. It's about providing feedback into the OECD process. So Ma is here from the OECD to take that feedback, and in many cases have proven instructive in telling us, well, why a thing is like it is, even if we couldn't see the sense of it. And once people see the sense of it, it becomes much easier to actually deal with. Why does it have to be like this? Why is the consensus there? And how do you deal with it? How do you deal with a comment from one country without then creating a new comment from another country, etc.? And it's about bringing together the stakeholder groups to try and improve how this development process runs, and sharing the best practice and learning. Next slide, please. So here we're going to have a go at the Mentimeter, just so we can get everybody settled in. We have 33 people here, so I'm hoping we're going to get 33 answers. You can see down the bottom right how many answers we're getting. We kind of will look at this, and if there's only 20 people in the first slide and we're waiting, then we'll sort of move on when we get to 20 people in the next slides, just so we see how many people are engaging with this part. We'll give it a little bit for everybody to log in. Okay. Website is in the chat, and there's a code that you need to put into a box when you get to the top of it. It's also at the top of the slides, as long as we're in Mentimeter. And in some cases, you will have to not just type your reply, but also submit it. It kind of depends on which kind of question it is. So we've got 20 now, which is about 2/3, which is usually what we end up with. Okay, good. We'll move on. If there are more people that want to join later, then they obviously do. So the next question is, which category best describes you and your relationship with the test guidelines? And you can see Bill's different pictures here. Sorry, Nicolina, I'm running over your slides here. I forgot to stop and hand over. Okay, so we've got a few more this time. That's good. See if we can get 23. No. Okay, good. It's a nice split. We can see we've got people from different sectors, which is good. Yeah. Next slide. Thank you. So the structure of today is that we'll start with presentations, as I said, from the experts from NanoHarmony and the OECD, who will present the experience they've had for going through specifically phase 2 and 3. There'll probably be comments on the end bits for context, but we'll try and focus on the phase 2 and 3 bits today. It'll be a mixture of Mentimeter questions and open debate, as I've said, and it is something where, as you think of questions when we go through the presentations, please stick them in the chat. It's the easiest way to capture it from 30 people. And either, if they're specific, we'll deal with them after each presentation. If they're more general, we might keep them for the back end when we've been through all the presentations. We'll probably try and get the people who put the questions in to read them out. Gives a better context than me reading them out. So you don't have to spell it out in great detail, just type your topic in and we'll get back to you. As I said, it is being recorded and it will be put on YouTube. So choose your language, so we don't have to put can only be watched after 9 o'clock on it. And the previous webinars are also available there, covering all the other aspects if you missed them previously. Next slide, please. So the table today, as square as the screen looks, we call it a round table. I'll try and share it. The experts that we have here today is Thomas Kuhlbusch, who's also the coordinator of NanoHarmony. So everything that goes right is to his credit. Everything that goes wrong is to our fault, and he'll talk to us about it later. Frank's going to talk about some work that isn't entirely NanoHarmony, but leans very closely up to it. And then Mar is going to talk from the OECD perspective. And I still haven't found the full slide set, so I don't know what's on the next slide. It might be here that we switch to somebody else and ask Thomas to get onto his slides, please. You're still muted, Thomas.
Yes, thanks a lot, Klaus, also for the hint that I was still muted. And I will now try to share my slides and get to the presentation mode, if I manage.
That's it. Perfect.
Very good. So
all yours.
Thanks a lot for the introduction, Klaus, and setting the scene, and certainly the topic on the experiences on test guideline developments and how we could improve or have a look into how scientists, regulators, industry, and all other stakeholders can work a little bit better together to maybe speed up the one or the other step of the process is something I think is really needed. And what I will talk here today about is the specific experiences we made when we had the test guideline development on particle size distribution. And just to introduce you a little bit into what was a task, and this may help to understand what were some of the problems we encountered, and so that we can see how we actually dealt with this, and maybe that we can learn all from it to make it better the next time. So the background of this test guideline is certainly related to REACH. It demands a physical chemical characterization of the nanoforms. Particle sizes have to be determined, particles, fibers in this case. And when looking into existing test guidelines, in this case, it was the OECD test guideline 110, particle size distribution, fiber length, and diameter distributions. It was from 1981. It does not cover the materials below 250 nanometers, obviously a problem for the nanomaterials. And certainly it does not comprise specific test methods for nanomaterials to actually have a clear look there, morphology-wise or in sizing as well. When working on this one, already this background, particle size distribution, the first thinking, and it was scientific as well as financial, as well as political thinking, which came here together. The definition of the term nanomaterial differs in the OECD member countries, often less than 100 nanometers, but there are some definitions going up to even 1,000 nanometers, like in some medical definitions. So we have to be clear whenever we set up a test guideline or we want to develop, we have to also look for their use in regulation. And here we certainly had to have a look of it for it. This test guideline development for nanomaterials was initiated and sponsored by Germany. And we decided to go for a new test guideline for particle size distribution in the size range of 1 to 1,000 nanometers. The next slide you have already seen, and I think hence it is not a surprise to you. There are quite a few steps. And when you look on the left-hand side, you see that the initial idea was in September 2015 to this test guideline. And from then on, we started actually to work along those test guidelines. And overall, even though we thought we were quite fast, from idea until the test guideline, it was now accepted by the WNT about 4 or 5 days ago. So it took nearly 7 years from idea until the test guideline was ready. And this is a little bit the background why we were thinking and looking into how can we speed up things if we want to keep pace with the developments, with materials, with regulation. Then we really have to be careful. We have to early think about what test guidelines are really needed. And we may have also to have a look in how can we prepare the test guideline in a more speedy manner. In this case, also for you to see a little bit, this test guideline is a very lengthy one because we had something like 12 different test devices for measuring the particle size distribution. So it's overall, it is more than 73 pages. It's pretty long for a test guideline. And it has a main section, beside introduction definition, certainly the important part, the scope and significance and use, which is a point you have to consider throughout the whole work along the test guideline. In the end, the test guideline has to fit to the scope and has to be usable to the scope which was considered. We have aspects of sample preparation, and then we have the description of methods, recommended materials for the validation of the test report, references, and appendix. And the methods were divided into a section for particles and a section for fibers. So this is the structure, 73 pages. It's quite a lot of text, a lot of science, which you have to do and which you have to fulfill. So it certainly means you have to have a lot of eyes into different directions. You have to have them open. And one thing is, when you want to build a convincing case, you have to really have a very good interlaboratory comparison. ILC stands for interlaboratory comparison, which means it's not enough that you have 1, 2, or 3 participants, that you have validated the robustness in one laboratory. You have to be able to be convincing in a scientific arena that it is robust and reproducible and everything what Claus was mentioning before. And therefore you need participants for the interlaboratory comparison. You have to attract participation, and you have to be sure that this participation comes from all around the world, not only one country, to build a convincing case with the scientific development of the background for a test guideline. And beside participants, you certainly need also the relevant scientific input. And this is, you have to have a look around who are the people being of interest and who are the people of relevance in your field, in your test methods around the world, and try to get them involved and try to get them somehow aligned with the test method. That will certainly help to build a convincing case. Another part which is quite often, or not quite often, which is sometimes lost when you are working for 7 years on something, and you have the whole validation procedure for this development ongoing. You see certain points which are interesting, certain points which are important for the one or the other reason. And it may well be that you lose your thinking along the line of the scope. What is really needed for the test guideline? Again, here also the scientific aspects. You have to think from the beginning until the end about the science which is needed to build convincingly a test method to be then accepted later on by the OECD experts and by the scientists worldwide. And this has to fit to the scope, to the reason why a test guideline was agreed upon by the member countries to be developed. And there are sometimes also points, and this is an example here, the single particle ICP-MS method is a method which some of you may be experienced or maybe not. It's a method where you use the mass spectrometer to actually size and identify particles down to, let's say, 10 nanometers, with some materials only up to 100 nanometers, and some go further down. And when we started, for example, to validate this method, in the end, we had not enough participants, and we had not the right material being really used then by the participants and being picked before. So in the end, we had a very limited data set, and with this data set was not very convincing. So in this case, as an example, it is that we decided in the end, even though the single particle ICP-MS is an important method to be used in various areas along nanomaterial detection and quantification and characterization, in this case, within the test methods, we had to put it into the appendix, and it is not part of the test guideline. So it is not a valid method to be used for particle size distribution measurement at this point of time. This another scientific expert already or aspect already addressed in the first bullet point is here expert meetings. To ensure not only the participation, but also the agreement with all the different scientists and all the different languages and different thinking, it is of very great importance to have regular expert meetings. And this not only with the OECD experts, but please also include or try to include international scientists. That's how we tried to do it, and how we then had the negotiation and discussion on the data interpretation and so on, to include international scientists to really have in the end good science, and good science in the validation report, which is a prerequisite for a test guideline to be accepted. By the way, the validation report is the report reporting all the work done towards the science background of a test guideline, where the results, for example, of the interlaboratory comparison are presented, interpreted, and analyzed. So these are scientific aspects along the development phase, but there are also some policy aspects. Or political aspects, things you have to consider as well. Different member countries have different areas of concerns, or have different interests in test guidelines or test methods. So it is quite important that besides your science, when you come along and come to the OECD and have a project proposal and later on the SBSF, and we are now in the phase between SBSF and the WNT, we have to be aware that different member countries have different interests, that this can be diverging issues. And even though us sometimes as scientists, we have to reach consensus on these diverging issues to really achieve that this test guideline will be accepted. So the different areas can be, in one example, there is one test method being developed by country A, another test method for a similar endpoint by country B, member country B. Both test methods, the member countries see as utmost importance to be part of a test guideline. They reach good interlaboratory comparisons for each of the methods. So here, then both methods may become part of the test guideline. But again, it has to be enough people to backing it up. It must not be only from one country. You also have to have the science in your view. So you can integrate them. It becomes very difficult if these two methods are not really ending up with the same, for the same endpoint with the same results. Both may be valid, both may be maybe robust, repeatable, and all the other parts, but it is not something as an SE, which you can trace back to SI standards. So let's say, for example, dustiness, where you have different ways of treatment, and with stronger forces or less forces, you will have different results of dustiness. So both methods are equally right and equally good, but they do not match to each other. And then you have to fulfill somehow, how does it fit into one test guideline? So this is something, or with the test methods, when you talk about particle sizes here, you have a test methods and another one, and one is on the optical size distribution, the other one is on the electrical mobility size distribution, the other one is on aerodynamic, and the latter one is the one we all know, which we can see with the microscope, the geometric diameter of the particles. They are all slightly different or totally different. So still we have to find a consensus, and we have to find a way, how can we integrate them into a method so that it can be then applied in all the different member countries for all the different regulations. And this is something what scientists and we as scientists are not really used to. We look, there's only one real answer and one correct answer, and the other ones are all incorrect. Not 100%, we can make different use. So here we also have to see, how can we integrate it? For the dustiness, for example, it can be the one method has stronger forces on the particles and stronger forces and tearing the particles apart, the other one less. It can be clearly that they do not match 100%, but they can be used to simulate different cases of exposures or treatments of dust, of powders. And in the other case, you just make clear that within the reporting, you clearly say, okay, we report optical size distribution, and then the regulation has to clearly pick up what kind of size distribution do we want to have. Do we need geometric size distribution? Then you have to pick a method which really fits to the geometric, or you can clearly deduce from that measurement a size distribution on the geometric side. But another one may say, okay, in liquids, we are fine with a hydrodynamic diameter, and then it can be slightly different. And then the definition is on this one given in the regulation. So we have to there also get the fit between policy aspects and the science aspect, and to bring it somehow together. Another phase before you actually can submit a test guideline is a writing phase. And one of the issues coming up during the test guideline of particle size distribution is the use of terms and definitions. The use of terms and definition can be very, very different between scientists, regulatory, and legal texts. So in the one arena, this word is meaning something, definition A. In the other legal arena or within the other member country, it has another definition. So while you are writing as a scientist and as a representative of an area you are involved in, for me in this case Europe, you have this in your mind, but you may not have in your mind what's going on in some of the other rest of the member countries. So we have to be very, very careful on the wording. One of the words which was very difficult in the test guideline for particle size distribution was the one of constituent particle. The term constituent particle is defined differently in the EU than, for example, by ISO. So whenever we are going to start to use this term in a test guideline, you either address a person with an ISO background or with a European definition background. And then it becomes difficult because the one determines or thinks differently about the same text than the others, and it is not clear anymore. And it becomes a touch of being involved or being very strongly linked to a legal text or to one of the regulations. So here it means that in this case, we had to define a new term, which is not used before by any other legislation or regulatory text, individual particle. was one term we then actually used and defined within the test guideline itself to overcome this problem and to make it clear that it is independent from any regulation, but still valid to be applied in the one or the other regulation. And this is something, again, as I said, a scientist may not be used to into this thinking, but here we have to bring the science and the policy clearly together. And it certainly is good if you have had that discussion already at the start of the project, that in the end, not the discussion starts, but that you can go back and say, yes, we have given enough time for the discussion on this aspect. We have this point of view, this and this and this, and you can then condense it and come to a conclusion. So this is how we can then tackle this kind of problem. So it's good to keep that in mind and to already address that and work on it before. One of the points which is always the part making it very difficult if there are some late issues. So which means if somebody finds out the interlaboratory comparison was very good, but it doesn't fit the scope, which means, hey, you have done something wrong. So you have to be prepared, you have to look, you have to really think that everything fits together. If those comments come, you have to be prepared, you have to know your science, and you have to know what you have done before with the meetings and so on, so that you can show, no, it works, hopefully. And it is important to keep in mind that the scope was agreed by all member countries. And so it is really important that the scope is backed up by all member countries still, and please keep in touch there. This is the second last slide. So I would like also to mention the validation report, which is the core of the science actually done within the test guideline development. The validation report brings in all together the background. Why did we do it this way and not another way? How was the interlaboratory comparison? What were the SOP used during the standard operation procedure used during the interlaboratory comparison, and so on. So this one is required for all TGs, test guideline. And it requires really good preparation from the start of the project. Why? It is because a lot of data will come together. In the test guideline, we have something like 31 to 32 participants with, as I said, 10, 12 test methods. If you start writing this validation report, preparing the data analysis too late and so on, it would certainly take too long. You have to really be organized and plan to go through all this discussion and prepare and analyze those data in time. And last but not least, the validation report, once agreed and discussed by all scientists and experts of OECD, WNT, it means it already has a backing that the science behind it is okay, that the core understanding is okay, so not that there will be not this type of discussion at the level of the WNT, then the meeting of the member country representatives, and it really helps to build a convincing case. Just from my experience, bringing a little bit closer and summarizing some general hints, it is important once you see a test guideline is needed to find scientific but also policy sponsors. I would like to call it this way. Certainly the policy is not really a sponsor, but you need the backing and of the member countries. So to have that, think that, and that is certainly done by your representative from your member country at the OECD, but you also have to keep that in mind. Inform yourself on interest in the different interests existing in the different member countries on the TG you want to develop, and try to include them and cooperate, and not to say, please do not say my science is better than your science. This doesn't work. Consider how to get different member countries and scientists involved. It's always here, OECD is working on consensus. Then include all relevant players, keep the OECD informed, and make your work understandable in simple terms and simple words. Please also write minutes of all the meetings and documents and document these meetings. This is important to make your work traceability, transparent, and so that the discussions later on are not coming up out of the blue, but you can say, yes, we have discussed it. We as experts came to this and that conclusion. People, once it comes to another level and another level, they start maybe again with the discussion. If you have it well documented, and if you can really then good show that this was not neglected, it really helps to speed up matters. And find consensus and prepare in preparation of the validation report from early on. Because if you can already agree on core issues in the validation report, then it helps to draft the test guideline and to get these things done. These were my experience, ideas, and thoughts towards the second and the third phase in the development of OECD test guideline. And I'm happy to pass on my words back to Klaus.
Excellent. Thank you, Thomas. I think there's some really good points in there. No questions have come up specifically, but I'm sure they will come as we go through to discussion. One of the ones that I could nod in recognition to was the thing about definitions, how much trouble you can get yourself in by using a word that only means one thing to you, but then clearly means something else to somebody else. The one we struggled with was a simple word like bulk, which meant, you know, at least 4 different things in the room of 12 people we had, but nobody really realized it until the discussions had been going on for a little bit, that we actually were meaning the same, but just not talking about the same. So, and I think what we found there was, you know, if you always describe what it is you mean, then it becomes clear to people if the term you've used isn't the one they would have used. And you alleviate the misunderstandings a little bit like that. Because it can be hard to find out otherwise that you're using it, that it's the word you're using that's upsetting the understanding. Good. I think we'll hand over to Frank, and then we'll take up discussions later on. Do you want to share? Thank you very much, Thomas. And Frank, do you want to share your slides?
I'm about to. Okay. Here we are. Please see my slides. Yep.
Perfect. All yours.
Great. Thanks. Welcome, everybody. It's my task now to go through another guideline and a guidance document development that is a little bit more historic. We started much earlier, but we had more or less very similar experiences. I also think that looking into what Thomas actually reported is that we had probably the advantage of being the first going through a nano-specific guideline development. And I think procedures are much more robust nowadays. And looking at Thomas' examples, there has been probably also a lot of changes in how deep actually those discussions go in the development of the guideline. Especially when I think about the efforts that Thomas had in the interlaboratory comparison compared to our efforts that we had. I will report on the development of the TG318, the nanomaterial dispersion, stability, and agglomeration behavior. And that was actually not a single isolated development, but this was part of 3 efforts. The dispersion stability, the nanomaterial dissolution rate, solubility, and the accompanying guidance document that actually should be covering both guidelines with enough guidance to actually also use or interpret the results in a correct way that comes out of these 2 guidelines. That actually developed a little bit difficult because the dissolution guideline was delayed, and since it couldn't directly feed into the guidance document 318, also the 318 was a little bit delayed. For the dispersion stability guideline, we had quite some work on the scientific background that was not directly related to a future guideline development. It was more basic science that we did in support of the Working Party of manufactured nanomaterials, but not with the primary scope of developing a guideline first. So in these years, we founded the scientific background for agglomeration, for dispersion stability of nanomaterials, and to test them in a background of environmentally relevant media. And so taking the existing science on how to investigate agglomeration or aggregation processes with colloids, usually, not nanomaterials, and translating this into something that is relevant for environmental conditions and also can be done in a normal laboratory that is not specifically equipped for doing experiments and analysis of nanomaterials. And there was, of course, a lot of discussion about how should the media look like. So what kind of waters do we use for this agglomeration testing to get an idea how the material would behave in many different kind of surface waters, for example. And it was clear that we couldn't actually use simply an EPA moderately hard water and then let it be with that. So we had to invent something new. So that was also then the parameter set and the resolution of our parameters, the time frame of the experiment that should actually show the process, but also should be doable in a working day in the laboratory, for example. Also how to do the sampling, and in the end also what kind of analysis can we use, especially in laboratories that are not equipped for nano-specific analysis. And that was very important in these years, because I just remember that we are now having a guideline on single particle ICP-MS, and it was 2008, still 2010 even 2012 in NanoDefine, where large criticism was exposed on us because ICP-MS is such a high effort methodology, so costly. Not every lab has that, and we were thinking ICP-MS is a standard option that you have in every lab, but that seemed not to be the case there. And that was also then actually becoming important for us, because we had to think about very simple approaches and not using the standard way, for example, using a dynamic light scattering for determining the increase of aggregate size over time as a parameter for agglomeration, for example. So, and also then possibly being able to directly conclude on attachment efficiencies, alpha values, and so on. So that was the major work in the TG development, experimental setup, reduce the parameters to a doable set, but still representing natural environment variability. Looking into what we can do with in silico models, not so much on the agglomeration side at that point, and also use an analysis that is not too complicated. We had then the implementation, also again 2 years for the implementation. That was a special thing. So there were some delays in there that were simply because of the layout and the funding scheme that we had that introduced some delay here. But also the fact that our second guideline on the dissolution of nanomaterials was delayed, hasn't been finished by now. And also this actually delayed the guidance development. So we started in 2008 when Austria actually supported the OECD sponsorship program for titanium dioxide. And we filed the first proposal to look into agglomeration under environmental relevant conditions to the Austrian EPA, and got a small project funded for 35,000 euros for 1 year. But there was also no continuation of that program. So we actually worked on this system with internal funds for some years. And in 2011, it was the time to present our final results and outcomes on an expert workshop in Paris to the OECD Working Party of Manufactured Nanomaterials. Then there was a kind of a break in 2012, so not much was happening in 2011 and 2012, so that we already anticipated that there might be no interest further on in these kind of processes. But then actually in 2013, organized by the German Environmental Protection Agency, we had a meeting on environmental fate and ecotoxicology of nanomaterials, and that actually stressed the importance of agglomeration processes of dispersion stability. And from this on, there was a SPSF drafted and submitted on the dispersion stability testing led by Germany, on the dissolution of nanomaterials led by the UK and Denmark, and also for the guidance document on both guidelines. Then also there was an end of 2013 a real research project funded by Germany, and that actually gave the starting point for really doing the necessary research and the necessary experimental approaches development for reaching this guideline in the end. And then actually it was quite quick. We had these 2 years or almost 2.5 years working on it. And we had from the beginning with the guidance also of the EPA in Germany that were actually already quite experienced in some OECD processes. We had the clear guidance from there how to proceed and how to do things. So they were our contact point to the OECD while we were the ones working towards the science. And that was actually a really nice teamwork. And I really enjoyed that. And I would recommend to everyone doing these developments to have a very close contact to the representatives, to the national contact points towards the OECD, because on the OECD level, there are things going on that you not, as a scientist, you will not be always involved in, and probably also don't have the time to always dive into these details. And that is really great to have a team that supports you here. But Thomas actually said more or less the same, I think. So we had our regular expert meetings. They were annually in this time because it was always in-person meetings. But it was always a very, very close discussion, very, very intensive work to find agreements on how these experiments should look like, how they should be designed, how they, in the end, should deliver a certain endpoint. And had also a lot of support in the final drafting of the guideline then. We then actually had also our last meeting in Paris to discuss the interlaboratory comparison. There was at these years a huge interest from laboratories to join this interlaboratory comparison. And so we didn't have that much problems to find people joining that and supporting us in this. That might be different today, but in these years it was quite easy. Then actually we went into the first commenting round and got our comments relatively quickly back. And that was first for me, because that was my first time doing this, that was quite a bummer to get about 40 pages of comments on my 9 pages of guideline. And since we had the case that the funding had stopped at that time, and probably this is specific for universities, Thomas, you might comment on that later, that we of course have a problem when these kind of pulsing workloads come in, while we already work on new and other projects that fully consume our time usually. So there was some delay in responding to this quite a lot of commentation on our guideline. We did so, we revised the draft, we sent it in, we got some comments back, but in the end in 2017, we finally got the approval and the guideline could go public. Important here, I would say, was from the beginning, the engagement of the expert group, an expert group that consisted of OECD listed experts and of course, international scientists that were prominent working in that field. We consulted them timely, we consulted them regularly, and we explained our work and we really listened to them. We really listened to the criticism and also the suggestions that came in to adapt our working scheme, our experimental scheme for the guideline. And we, from the beginning, actually searched for an agreement within the group, what actually I think made the commenting round much easier for us, because most of those that were in charge and commenting on the guideline already were informed. They knew about how the development had been going on, where were the points where we made decisions, and that actually made it very much more simple. It's mainly the experience that Thomas also had, and also the recommendation from my side, include those people, discuss your problems, make decisions together. It is not the case that that thing that you do is the best science and the other science is wrong. It is searching for a compromise and for something that is really valuable in the end for everybody in the OECD world. As I already said, for us actually it was difficult because we didn't have any permanent staff available that we could actually give the job to. For us, it was this pulse workload with having nothing on the table and then having commenting pages within some days or weeks to do. That is difficult to integrate sometimes, and that was actually for us not foreseen in that case. Now we have also other developments going on where we actually definitely foresee exactly that in the commenting rounds we will not have staff available from the project left over, but we as a permanent scientist, we need to take care of that and also make plans for this. The good practice that I have learned actually is that we actually abandoned the idea to produce now the best possible assay that you can produce that contains everything that could actually happen. We had this discussion long in the group how we should actually select the pH ranges for our test, and we made a survey of surface water parameters for pH, for other for anions and cations to really select the range, the most appropriate range of pH and ionic strength and so on for our testing. And then in the end, it was just the last minutes of the meeting, actually, it was, yeah, for harmonization issues, we would suggest to use pH 4, 7, and 9. And that is not really, especially the pH 4 is not really environmentally relevant. Also the 9 is a bit far out. And it was much broader range than we had actually suggested. So we had hours of discussion about our range, and then within minutes it had been changed to something totally different that I think also has no environmental relevance. But for harmonization issues with other guidelines, that made sense, and it was agreed on, and so we did this. We still have discussion about this, and there are some people that don't like it, but that is something that you have to compromise on in the development. You need a strong support from your team of regulators. In our case, that is the team from the EPA in Germany, Doris Völker and Katrin Schirn, and I really like their support over all the years in this work, and it's just a great teamwork with them, and search for those people that are your link to the OECD. And of course, your national contact point. Include the OECD experts and your scientists from the very early beginning of the development, reach consensus on the really important points, document your decisions that you make when you change things in the development and so on. Also search for experts from as many countries as you can get, because that makes your commenting rounds much easier, and also you're producing a guideline in the end that is much more accepted by the whole OECD world, and yeah, it makes your life easier, as Thomas already said. In the end, accept it as a collaborative process between scientists and regulators and industry representatives all over the world, in the OECD world, and seek for the best compromise. Everyone has a right point here to come up with something and try to integrate this into the guideline, and don't grade anything in terms of it is industry related or it is scientific related whatsoever. Everybody has the same voice here, and try to find compromise in the collaborative process. The Corona lesson that we had actually is meet more frequently, use the online meeting possibilities. We got so used to it as we do it today. We got so used to these tools over these years, and just keep it on and make your more frequent meetings, even also with subgroups of your expert panel. If it comes to certain questions, just contact them, try to find a 30 minutes, 1 hour slot somewhere, and discuss your points that you have in your development. Barriers that we saw. For me, in the beginning, it was totally, it was a black box. OECD was not really clear to me. Also not how the procedures go, who's responsible for what, where do I have to pay attention, what is the timelines, and so on. So that was something that was absolutely not clear. It's also not easy to obtain. So you have to ask people and go through and train yourself on that. I think these workshops like NanoHarmony does it now is extremely valuable and helps a lot. And I'm thinking about how could NanoHarmony probably expand this to the whole OECD developments. So not only the nano-specific ones, but also others that develop OECD methodologies could probably really profit from these workshops and the recordings. Another thing that is clear for me was that without a really clear funding plan from, let it be EU or your local authorities or even cross-country funding, always when there was something going wrong, it was a problem with the funding or with the continuity of the staff on the side, so that people changed, that responsibilities changed, and there was not really a focus and real effort put on the development, because simply it was something that had to be done sideways or in parallel to other developments. And funding actually is the one of the major issues here in really driving something through to completion. Then the decisions on the design of the experiments, the design of the analytics, the decisions about complexity of the testing versus the relevance and the implementation later on. There would be, I would like to see a higher engagement of regulatory bodies, more feedback from ECHA, for example, but also from other international regulatory bodies, because ECHA is only representing the European part in OECD. That was a little bit, that could be improved actually. Of course, more frequent consultation of the expert group that is now possible via the online meetings. Less result presentation in the groups, but more consultation on the pitfalls, more agreement, making really clear what we are talking about, deciding on terms, as Thomas already said. And there's another thing that I found during the dispersion stability development. While we aim to simplify the whole thing and to develop it to something that is really doable in a commercial lab, the research of course went on. So we decided to use a very simple analytical approach and abandon anything that has something to do with dynamic light scattering, because such a complex instrument. And when the TG was ready, almost every laboratory had a dynamic light scattering instrument, and I'm getting requests, not frequently, but from time to time, by companies says, can't we do this with a DLS? We can do this easily, but this is not in the guideline. How does that compare now when we report DLS values? And also the HOMO versus the heteroagglomeration. We excluded the heteroagglomeration, although it is very important and the more realistic parameter, but there were no protocols available and very, very little literature data. Only in nanophase, we were able to really concentrate on the problem of heteroagglomeration and develop it within 3 years, 4 years, into something that might be possible to validate and standardize and bringing into a guideline, or at least expanding the guidance document. In the commenting rounds, actually, we saw sometimes really contradicting requests, even from the same member state. The one comment actually saying that we should add complexity and consider some other parameters and influences and processes that we already had actually excluded, because a minor effect and a seldom to see effect and too much actually effort on the experimental side to include this. And at the same time, the member state or the member actually was asking for lowering the costs. So giving you actually a problem in hitting the target, because it's simply contradicting. Also, we got repeated comments then from different member states in the 2 different rounds. So we had to explain the same thing that we had done actually already in round 1 in a slightly different way than in round 2 again. That was duplication of effort in that case. And sometimes you found that people were going back to really basic research and science like colloid chemistry to discuss very, very specific issues in terms of agglomeration models and so on. And that had been discussed 100 times already in the expert group, but had to be again brought up onto the table in the commenting round. And that actually showed me that probably some of the people that were doing the commenting should have been invited to the expert group, or nowadays we would probably record our expert group meetings and then simply show actually the discussion that we had on this. And another obstacle and barrier that we see currently is that these developments contain a limited amount of basic research and also a limited amount of possible publication output. And that is, at least for a university, not an easy thing, because our people that we employ in the projects as project scientists are on their career way, and they actually are searching for groundbreaking research and publication output that is not so much possible in these projects. And this is also what we see as new challenges in the development on our current guidances and guidelines on environmental transformation, dissolution solubility, dissolution rate, and heteroagglomeration. The career opportunities for our project staff is relatively limited. Little groundbreaking research, basic method development, a lot of effort into ILC measurements, repetitive activities, and limited publications. And that actually piles up currently. It was better the years before, but currently we see that we see really little interest from postdoc researchers to join these projects and do these 2 years of guideline development. And we also have a high volatility. When people started, actually there's a high risk that they break off after some months and go to another research group where they can pursue their basic research and increase their publication output. Also, we cannot employ any PhD students on these projects because also they are forced to publish. Current solution for us here is that we are looking for candidates that have expressed their interest in regulation. So we directly ask them in the interviews and also tell them the background and ask them if they are interested in going towards regulation and getting through these projects experiences on regulatory work on the OECD level. What for those postdocs actually is a really good thing. Thank you very much for listening. I repeated a lot of Thomas actually, but that's probably part of the game. And looking forward to your questions and the discussion.
Excellent. Thank you very much, Frank. Yeah, Sean just saying he's joined. He's missed all the initial bits, so we'll see what question he comes up with later, maybe. I think it's the bit you've had in there a couple of times of how we make room within a test guideline for accommodating the technical development that goes on both in the lifetime of the development itself, but also in the lifetime that a guideline has to be in service, is something that is really important. I don't know quite how we work it in, but that would extend the life of a test guideline so much more. There's no specific questions. There's one from Rachel. Academic involvement in the OECD. It's about motivation. We'll pick that up at the end after Mars slides. Maybe Mars got something in there. You know, you get 2 pieces of chocolate or something. I think it is really about which institution. How do we get institutions to see the value in the impact that test guidelines have? It's not something OECD can do themselves, apart from maybe an authorship kind of thing. Ma, do you want to share your slides, and then we'll get to the general discussion afterwards? Thank you very much, Frank. Excellent.
Now, can you see it?
Perfect. Okay. All yours.
Okay, well, thank you so much for letting me to present the OECD this part on this, and I really appreciate the points that have been made by Frank and by Thomas, because I think I can only agree with them, all the points that have been raised, and I can only sympathize for the frustration they have gone through. I've been near them in this process, and I can fully understand, and it's something that indeed needs to be improved, especially this constant round of comments and sometimes very frustrating reiterations, and sometimes this incoherence within a single delegations to provide comments. But I think the feedback is well taken, and I think something that we have to make sure that we can convey as we move forward. So what I would like to share with you today is just a quick reminder. I'm not going, I mean, this has been said several times, but I think it was important just to include this slide in the presentations, because as they're going to be made available, I just wanted that the slides make sense for themselves. So the OECD task guidelines are very important, because at the OECD we have this mutual acceptance of data, which is legally binding instruments across OECD member countries and other MAD adherents. And the real idea behind is that you test it once and it's accepted everywhere, so you don't have to do repeated tests. And this is the rationale for being so perhaps reiterative in these commenting rounds, and to make sure that we have everything done on a consensus basis. And I will, the slides are going to be made available, but I just invite you to see this very short video on how this mutual acceptance of data works, because it's quite illustrative. So why I cannot see, okay. So something that I might want to stress about the task guidelines is that there are different outputs of the task guidelines program. So obviously what you have seen, and Thomas and Frank have presented, are the task guidelines. This can be new task guidelines, updated, corrected, or simply a discussion on how to delete those that have been out there for quite some time, so that became obsolete with the advance of science. But you might want to consider, if you want to take this adventure to other type of documents or tools that can be supporting the task guidelines program itself. So on one hand, these guidance documents that will really contain all the information that you wish to give to the study director, so they can actually apply the task guidelines in a proper way when using, for example, in this case, nanomaterials. So it could be an existing task guideline that has been made 15 years ago, but you actually know that there are some specific modifications or specific guidance that you might want to suggest for the use of an existing guidelines. And this might be a little bit easier, because you don't have to go through all this validation process that Thomas mentioned. Then you can obviously do some validation and peer review reports to establish the foundation of a new method. So that's going to be like a task guideline proposal that you might want to submit. Or you can also do a detailed review paper just to explore a new area of testing. So those are different options if you want to go through this process. So we have the national coordinator. So it's the working party or national coordinator of the task guidelines program. And those can be one or more representative per member country. Is really the governance body for the task guidelines programs. They are the focal point, and something that is quite important to remember is that they will be representing the views of their countries, the legal position of their countries. If you, in doubt, you can go to this link. You have the full list of national coordinators from every single member country. And I think the best, as it has been mentioned before, is really, these are your best friends, because the national coordinators will not only help you to make sure that you can fine-tune your project proposal, but they also have an overarching views of other existing task guidelines that have been published, but that also, and you can see those online, but also all those projects that are on the way that might be impacted by your proposal, or perhaps your proposal can be integrated in something that is already on the way. This WNT program also includes the participation of industry that provides a lot of scientific feedback, and animal welfare organizations such as ICAPO, because of course animal welfare is one of the big trends in the work of the national coordinator, is something that is getting more and more important over the years. The national coordinators meet only once a year in April, so this is quite a very strict timeline. They are, and at these meetings, like the one that we just have last week, is where you're going to approve task guidelines, a guidance document, everything that you need to publish is going to be discussed there. They're going to be discussing the new project proposal, so the standard project submission forms. And they're going to be fine-tuning, sometimes addressing some late comments that might have come up in the latest process of the approval for task guidelines. And they will try to exchange information also on things such as whether there are ways to improve the flow of communications, etc. There is a strict timeline, as I mentioned. So every 15 November is the deadline for submitting a new project proposal to the WNT. If you miss this deadline, that means that you will have to wait a whole year in order to submit your project. And then every year for those standard projects, projects that are already on the way, the 10 March is the deadline for submitting documents that are going to be submitted for approval to the WNT at their meeting in April. But something perhaps to reiterate is that the comments to an SPSF can be submitted between December and April. So you need to be aware that you might have to be quite reactive, in particular in the last couple of months before a meeting, just to provide supplemental information, so you can convince if there is a delegation that still has doubts about your proposal. One thing that is discussed at the WNT is also the support that you will have regarding your new proposal. So national coordinators need to provide comments, need to express whether they support your proposal, but they also need to express whether they're going to be assisting with expertise in the development of your proposal. And this is why this communication that has been flagged by Thomas and Frank on looking around, start discussing with first with your national coordinator, but also with other countries that can be on the same page and have the same interest than you, so you can have your project proposal approved. And every year, just to say, we can have between 10 and 25 standard project submission forms submitted to the WNT. So you can imagine that when we have too many, well, we try to find the balance between the capacities of the different expert groups to provide feedback and the needs from a regulatory point of view. So all these things come into play. So the SPSF is really it's really like the roadmap of what are the type of questions and issues that you will be required to answer, and where the national coordinators are going to be quite picky about. And obviously one of the most important is whether there is a regulatory need. So you need to go back to your legislation. So if you are in Europe, you're going to go to REACH, but likewise for any other country. And you will have to bring forward what is your regulatory justification. So it's not because science is great and because we have a new method that is more effective that is going to be sufficient. You really need to justify that this is a regulatory need, because ultimately it will be part of the mutual acceptance of data. Okay, you have to justify whether this is like your country is the only one interested on this, or whether there are more countries that are going to be supporting this endeavor. you have to justify whether this stage G is broadly applicable, and also the type of methodologies that you're going to be proposed, because something that you might want to consider is that you want to make sure that you avoid any monopoly for laboratories in doing a test. So if it's something very complex to perform, then you might be asked to wait some time before you come up again with the proposal. And this has happened in the past. You have to justify, obviously, all the timeline, all the funding, whether you're going to be organizing some validation test, and then some additional resource related questions, such as whether you need an expert group, whether you will be needing support from the Secretariat to organize meetings, etc., etc. So this is like a really like the checklist that you need to go through, that you need to try to answer as detailed as possible, provide sufficient reference to publications that make sure that the WNT is comfortable, that the research is mature enough in order to be incorporated into an OECD project. And again, just to reiterate, not all the science can be used in regulations. So something that you have, this is where the WNT can be extremely helpful in providing feedback. So just contact your national coordinator. You can imagine, and as it has been said before, that once you have an SPSF submitted to the WNT, this is just the beginning of a very long and extensive consultation process. First, we will be asking you whether you need an expert group to support the work. In the case of nanomaterials related projects, we try to reach out both to national coordinators and also to the heads of delegation of the WPMN, just to make sure that we have a very good coverage of expertise available between nano expertise and other test guidelines expertise that are out there, to make sure that we can have like an horizontal screening when we provide different comments in the different stages of comments. And then it will be to the lead country and the expert group to work on technical aspects of the test guidelines and to build some consensus. And for this is what Frank was mentioning. I mean, we need to have some meetings within the expert groups of nominated experts from the WNT and the WPMN, because this is also like experts that have been nominated have clearly a very close communication with the national coordinator. Once we see that all these, the comments are pretty much, that have pretty much been answered by the leads, then we feel that we are in a comfortable zone to go to a WNT review. And in this case, the type of comments that you might get might be different. And this is also part of the, sometimes the backlash to the scientists that have been behind the drafting of these test guidelines, because they're going to take into consideration other issues. They're going to take into consideration, for example, whether animal welfare have been taken into account properly. And the discussion can go quite rough, and because you have to justify whether you have like sufficiently statistical weight for the proposal that you're making, but at the same time, you cannot propose something that is going to be, for example, or more expensive or use more animal tests than a methodology that is already there. You have to make sure as the glossary and the terminology that you are using and that was fine-tuned in a perfect way by the expert group is not contradictory or is coherent with some specific terminology that is used in another test guideline. So something that you have to cross-check. And we have had that surprise that at the moment of the WNT, we realized that there are a specific concept like Claus just mentioned on bulk, but it could be on solubility, on dissolution, and suddenly all hell break loose And this is where your national coordinator are going to be your best allies, because they're going to fight and they're going to try to find consensus with their peers within the room, without the experts in the room, and to find and approve an agreement so the document can be approved and finalized. So the timeline can be 2 to 5 years. You heard from Thomas that it took quite a long time for their task guidelines to be approved. There are other guidelines that take less time, but they are less complex. And this is something that you also might want to take into account. I think that the trend in the test guidelines program is that we are having, we have to make sure that the old test guidelines are curated. So we need to check on obsolete methods, et cetera. But also the methodologies that are being proposed are more complex nowadays. There is a lot of work to avoid ambiguity and to increase the clarity of test guidelines. And this effort obviously translates sometimes in an extra round of comments that was not foreseen at the beginning. So the WNT will approve the documents in April. So that was, as I said, last week. And congratulations to Thomas on that important test guideline. And now what is going to happen is that all the documents will go to the Chemicals and Biotechnology Committee, which is the parental body of the national coordinators. And then they're going to be endorsed and they're going to be published, and they should be publicly available at the end of June, beginning of July. So they can be part of legislation and be used in a regulatory context. So who are these mysterious commenters from the expert groups? So they are nominated by national coordinators, by BIAC and by ICAPO. In the case of nano documents, we have some joint nano test guidelines group, for example, on physical-chemical properties, on ecotoxicity and fate. And we have some other tailor-made expert group for specific projects on the way. For example, we have one on the toxicokinetics for nanomaterials. And so this, in the case of the last one, the one on toxicokinetics, those are really other groups that will exist as long as the project is on the way. And then you have the other ones like the physical properties or ecotox and fate that will be responsible for overseeing all the projects within that section. So for example, the physical chemical properties, they had, I think there were like 7 or 8 projects under their scope. And the idea is that experts that providing comments have a good overview of what is going on in the other test guidelines or guidance documents under development, and that we can make sure that we can ensure consistencies and avoid either conflict results, or in the case, for example, of the terminology, that we are using different terminology when preparing the documents. We have some advisory groups, but those are not as specific to nanomaterials. And all the work that is being done within the test guideline has to be shared within a restricted site that is only accessible to nominated experts. Something to mention is that some lead countries in developing their proposal, they organize their own expert groups. This is something that is the prerogative of the lead country. And so they can get sufficient support before the document is circulated to the OECD. In the case of NanoHarmony, for example, this has been a great advantage because you have a whole community of scientists providing the feedback, the necessary feedback, and providing the resources for the development of these documents. And then by the time it goes to the expert group, is something that is more digestible and easier to take account. So I just wanted to again stress what Thomas said. If you're going to do a test guidelines, you have to consider validation and standardization. So this is the type of information that a national coordinator will be looking at. So you really have to have the relevance of the methodologies that you're using. You have to make sure that it's predictive, that is reproducible. And this is where the amount of laboratories that are participating is going to be quite important. And I think just for statistics, the test guidelines on particle size and size distribution that was led by Germany, I think it has like a record number of laboratories, which I can't remember if it was something around 30 laboratories, but that was like, something that is unseen. Normally, if we have 10, 15 laboratories, but that are distributed across the globe, that will be sufficient. Some quick reminder, as Klaus just mentioned, always respond to a regulatory need. If it's not directly responding to a regular need, don't bother, don't bring it to the OECD, because it's not going to be, it's quite likely that it's not going to be accepted. You have to make sure that your proposal is for robust and reliable methodologies for governments, so they can actually base decisions on approval of chemicals within your nation. You have to be mindful of animal welfare considerations, and obviously something that might not be scientific, but just be mindful of the economic implication of the testing, and it has to be broadly accessible as possible, okay? Lesson learned, so as it was mentioned, test guidelines are done on a consensus basis. Sometimes we get in a loop of a comment that was mentioned, and then we have a discussion, a bilateral discussion. We try to accommodate those comments, then we go to an expert meeting, these comments come back. Sometimes it's better to give up on that. If there is no consensus, sometimes the easiest is just to delete it if that is not going to be something that is going to bring too much weight or affect the test methods that, that you are proposing. At the end, what we really want is that we have to preserve mutual acceptance of data in light of diverging requirements in member countries. So this is something to keep in mind. For the SPSF, it's really advisable that you don't be too ambitious, be very clear, be very succinct about what you're trying to propose. I think Maria Luisa from Spain, they just made it through an SPSF that was suggesting some very specific additional information to be included in an existing guidance document. And this is very straightforward, so it's something that can be done also, but try to avoid changing the scope of your document in the process, because then it might just open up Pandora box, and then we will have to reiterate and go back to the national coordinators for agreement if something else has to be touched that has not been put forward since the beginning. Another thing that I just wanted to mention is the national coordinators really have all the institutional memory across the program, and they will have a very good overview of all the different expert groups. The last layer of comments that you provide, so when a document goes from an expert round of comments to a WNT round of comments, national coordinators have to agree or disagree with the expert comments. And for the lead, it's really important to have that backup, because if the national coordinators agrees on the comment, that is something that obviously will take more weight than if there is a disagreement. And again, the reason is because they are looking more not only about the science that you are presenting, but also at the policy implications of the methodology proposed. And there are some information on YouTube that you can access regarding the test guidelines program and the validation principles that have been done by my colleagues. Obviously, you also have NanoHarmony that have made quite a number of webinars regarding test guidelines, and I really appreciate this effort. I have learned a lot with NanoHarmony in trying to better advertise our work and trying to make it more transparent and accessible to the outside work. So that's a big thank for you. And I think that's it from me.
Excellent. Thank you very much, Ma. And yeah, my apologies for my introduction being too long, so we're slightly late here. But I'm hoping, as I said in the chat, that everybody's got another 10 minutes for the discussions. There were some specific comments in the chat, but I think they've been resolved within the chat. So I think one of the general things that was mentioned and possibly isn't resolved within the chat is this thing of how do we retain the motivation of the academics to be involved for this long time. You know, we have to get new people involved and understand it in terms of the young researchers, but really we have to somehow get some authorships and impacts in there that is recognized by the institutions that we work for, which for people like Thomas is probably a lot easier than for people like Frank. To take two end members of the audience here. But I don't know, Ma, if there's any, you know, when we work for people like EFSA, there will be a report at the end that says, you know, these people are the ones that contributed to this guidance or whatever.
Yeah, I think we have, this is something that has been discussed within the OECD and something that I will need to report back, because I know that, for example, when we did the inhalation toxicity test guideline, when we review it for nanomaterials, the original test guideline had a lot of thanks to, and then you have all the list of experts. And then by the time we had to do the nanomaterials one, that section was removed. There was a decision to remove it, but I think it's something that has been mentioned several times. And something that is currently under discussion. Yeah. So, but something that I might need to get back to you on that one. Yeah. Because it's true. I mean, it's done, all the work done at the OECD. At least, I mean, we don't pay anyone to do the work, and everyone is doing it on a voluntary basis or paid by their own government, et cetera, et cetera. So it's something that obviously needs to be recognized.
I don't know, Frank, if you have any ideas of how a recognition scheme could look that a university would accept.
Yeah, it's always difficult because every university has its own evaluation scheme of scientific output. We have pressed more and more that this outreach to public and public engagement is more quantified or more quantitatively taken into account. But there's some kind of reluctance because they don't see how they actually should, how they should quantify it. What is an OECD? People don't know what an OECD guideline is actually, and they can't actually evaluate it in terms of how much scientific output you have or how much effort you have put in that. It's difficult.
Yeah, but definitely something to work on in terms of keeping people involved. I'm just trying to. Yeah, there's one from Fred. Do we maybe, given we are over, do we want to run through the last few slides, Nicolina, and then hopefully we can, when we're at the last slide, we can take the general discussion back up then.
Sounds good.
There's only like 3 or 4 here. So if you log back into Mentimeter, then Nicolina will run you through it.
So you can, just go back to the same page you were earlier or log into menti.com and use the code 59842603. I'll just wait until we have a more substantial number here.
Good. To some of the things I'd like to discuss afterwards is how one of the things I've noticed in Thomas's and slightly also in Frank's discussion was that you mentioned scientists and policy, but we didn't hear much about is industry involved, and that's sort of also borne out in Fred's question of at which stage do the non-GLP labs I guess my side of the question is where are the CIO labs when we're running through the technical development in terms of making sure they're involved in the ILCs, and we kind of end up getting that strive for simplicity worked in as early as we can. Okay, so Nicolina.
Yeah, so I just open up the first question. So what level of direct experience have you had with the OECD test guidelines program? And here, I won't read all of the answers, so not too long, but yeah. And we'll wait. I think we had 14 people logged in, so I'll wait until we at least approach to number 14.
Perfect. Frank, I don't know whether you want to comment. I can see you have to leave for another meeting. Do you want to comment on how we would get industrial labs and CIOs involved within the phase 2, really?
In principle, it should be easy. I think it's a question of information. So knowing actually which labs perform on a regular basis guideline testing for OECD, and then actually they are usually interested in participating in it. And for example, in the TG 318, we had one participant from a large German company that is doing commercial testing. And I think it's a question of information. Labs are interested. So, but that would probably require that someone actually digs out which labs are actually actively working with OECD methods and then keeping them in an information loop like a newsletter or something like that.
Okay, good. And I think this is the most informed audience we've had in terms of constant engagement. I think mostly we've had about 3/4 of the people having very little or intermediate engagement, but we have quite a strong cohort of people today that has constant engagement with the TGs and active programs here.
I will go on to the next question now. So do you think the current phases 2 and 3 process could be more efficient or quicker? So simple yes or no question.
I'm interested in the how.
Yeah, I'm interested in the which bit, because I think the way you presented the OECD bit is quite set, and it has to be quite set in diary terms because you need to engage quite a lot of people and you can't do it runningly and all the time. So to me, I imagine the bits that we certainly from within NanoHarmony can write the best pieces of guidance on is how those soft bits in the middle take 2 years instead of 5 years to get the documents right so they're ready for WNT.
So it seems we have a majority for yes.
Yeah.
And now, do you think that there are difficulties with recruiting labs to support an ILC for a test guideline or guidance document? So.
And while that's going on, Fred, did I interpret your question correctly that it was, obviously there's universities involved in the ILC, but should it really have more GLP labs in there to make sure the protocols are tested in a hard environment?
I think there's 2 parts. One is, do you really need to be GLP to demonstrate reproducibility? And the other is, are there some requirements that an academic lab or a company lab that's also not GLP should accept or add to their normal procedures that would make them more acceptable for this purpose. So, because you have the experts at the lab level, but the experts at the OECD level are probably experts in toxicology or other topics than on solubility, for example. So where do you draw the line and make it hard GLP versus soft GLP?
Yeah. Can you comment on that, Ma, in terms of, does it affect how the evidence is seen when the validation goes through?
Klaus, might I just add, Thomas said 30 labs. How many of Thomas's labs were GLP?
Okay, well, Thomas has got his hands up, so you can answer that one and something else. You're muted, Thomas.
Again, I was muted, so it took some time. Fred, as far as we know, at least during not all of the 31 or 32 labs which were involved were working according strictly GLP. They had all the regulations which you have to have for a good laboratory, but not were not always in this way to the end certified. I think for testing reproducibility for these type of intercomparison, and depending on which type of method you have, it is possible within the interlaboratory comparisons to include labs which are not certified. The use and the application of a test guideline in the legal area so that it gets accepted by MRD, then it has to be done by a certified lab. Otherwise, certainly you can use those results, but or use those test guideline, but you don't need this then the certification of the lab.
Thank you, Thomas. Ma, did you want to comment or?
Can you repeat the question? Sorry, I was also answering.
Just in terms of whether there are extra pieces of evidence that the academic labs have to put in or adopt in terms of recording structures for the data really to be have the weight it should have when it goes through the validation round.
Yeah, I mean, in that sense, I think it will really depend on the nominations from national coordinator, and I think, I mean, if the national when we sent a call for experts, we try to provide, we provide the SPSF, we try to provide as much information so the expertise and describe the expertise that we're looking for. Yeah. So I think this is where national coordinators are really key in identifying proper expertise and a good balance of expertise.
And do they also, do you also use the national coordinators to get the candidate labs for the ILCs?
So what we do for the calls for laboratories, we send that to the national coordinators. Yeah. In the case of Nano, we also send that to the working party on manufacturing nanomaterials. And then we also put on copy all the group of experts that are related to that specific task guidelines to try to reach out as much group as possible. We do have a set of a distribution list for laboratories that have been participated in different task guidelines. So they are like, let's see, like the common suspects for this. So they also receive the calls for laboratories. But again, it's something that if you're not used to the way of work with OECD, for example, we got a question in one of the ILC calls that we did, I think it was for the solubility and the solution project, and it was like, okay, so how much I'm going to be paid? And it was like, well, nothing. Actually, I mean, we OECD, we cannot pay you for anything. So it really depends. We will provide the materials, they will provide the SOPs, etc. But that depends on the individual arrangements within the proposal, the lead country.
Yeah, and that, I guess, is what's in the blue square here, that the people that have experienced difficulties with this is because where does the funding come from for the ILC? And that's certainly something we've touched upon before in NanoHarmony, that the sooner you can bring that funding online and have a realistic expectation of what that funding level is, the the faster it'll go, because this is where some delay in that middle phase happens in terms of recruiting the labs and getting it done in a timely fashion.
Yeah, and I think the experience with the COVID was, I mean, that was another experience, right? I mean, we called for labs and then labs were closed. So this is something that when you come from when you're funded to do some work, you continue reinventing a little bit and try to find some solutions to cope with this lack of progress, but it's so much that you can do, and then you just have to press all the ILC that you were foreseeing for a year, then everything you need to do it in a year, because otherwise you will lose your funding. So this is the part that is, but it was, let's hope that this was really like a case that will not be repeated.
Yeah, I think we all hope that. Good. The next slide, Nicolina.
So have you experienced issues in reaching consensus during the development of a test guideline between member countries? So
there must be some good stories here in terms of what causes these issues when they happen. And is it something that apart from definitions like Thomas discussed, are there some common mistakes that causes issues around the consensus finding?
I mean, but you need for the development. I think the clearer you are in your project proposal, the easiest is going to be later on. I mean, we had I would recently have an experience with one of the SPSF that went submitted to the WNT in 2014. So you can see the difference between an SPSF that was submitted in 2014 and one that is being presented now in 2022, or the one that was presented, for example, for the particle size distribution in 2017. But so there is a maturity that has grown over the time, and I think this is part of the. If I can go back to your phase one, which is the pre-SPSF WPMN segment, where there is an expertise that has grown over the time, and the clearer you are, the easier it is to get agreement. Then, if it's not scientific and it's not added value for the test method, just take it out, because it's going to be a conflict with someone. I mean, there's no way you can go through that. No.
So the less opinion and the more fact you can make it, the easier it'll go through.
And justification, why you're doing it. And something perhaps just to mention is like the fact that you receive 40 pages of comment doesn't mean that you have to agree with them. This is also, the expertise is on the lead, and the lead can actually say, no, we are not using this or that because, and that's something that is also, it's part of the dialogue, I guess, that needs to be put in place.
Yeah. And as long as you're evidencing that rejection, then you could still reach consensus.
Yeah.
Okay.
Where would you expect future test guidelines to emerge from? And here you have to rank the answers in a descending order.
And then press submit.
Yes, always press submit. Yeah. Good point.
Good. So there's a few bits in the chat while people just fill this one out, because that usually takes a little bit longer, which is really around the terminology and how we reach harmonization. I think the biggest problems we've seen is not necessarily, there is some harmonization within the OECD, but it's when it differs from that found in ISO. I think you mentioned that, Thomas. Where did you work from to kind of establish that terminology? Did you define it within your test guideline, or did you go somewhere?
It was first within the test guideline, and it was said we should follow the ISO terminology. we clearly found in the one or the other case that the terminology, not in all cases, really fit to the scope and to what we want to express. So we started already there. And as said, in some of the cases, it became evident that if we use a term in this way, it would interfere with the understanding in some other regulations. So in the end, it was a discussion within the expert groups and finding a good way and solution. And I think it took something like 2 or 3 extra meetings, sometimes in smaller circles, to identify and find out solutions. And in the end, in the large circle again, then to agree on a way forward there. So that's why we have then in the test guideline a section in the end, which is on the use of terms. And it's specifically the use of terms for this test guideline. And it's not meant to be a general definition. And I think this is, as we also agreed with MAR, it should be as much in agreement with all the other test guidelines on nano. But it certainly has to be clear for this test guideline here.
Yeah. And I think that's also, thank you, that's also reflecting in Mar's reply in the chat that, you know, each test guideline will hold a set of specific definitions for that test guideline. This set of answers here suggests that there will be demands from regulatory bodies for new test guidelines that will need to be fulfilled, and that most of it's going to arrive from within funded projects in terms of technology being developed. So there's obviously an easy connection between those in terms of regulatory bodies can influence funding agencies to fund projects that do, but that also takes time. I think the other element is the pink bar, you know, what are industry needing? And there it's probably harder because for industry to state those needs, they need to say what it is they're developing that will need new test guidelines. And we have discussed that before in terms of a trusted environment where people can say, well, these things are being developed, therefore these things need regulating. I don't think that's really, that's probably more for phase 4 and phase 1 than phase 2 and 3. I think the bit in here that I find important is if we take the stuff that's coming out of funded project, that will be cutting edge science. And a bit, we discussed it a little bit in terms of how do we make sure technical guidance that is written is open for improvements in that measurement technology, so that, you know, you said, Thomas, you had to write the single particle SPMS into the annexes. By the time you were finished, it's got a nearly in every lab kind of thing. Is there a method for either reviewing the MAR, or is there a better way for stating the principles of what must be measured so that any new emerging measurement technique that can fulfill those requirements can be used? How do we write the test guidelines so they are as future-proof as possible?
I think, yeah, I mean, I think what I will do is, I mean, the test guideline itself, you have to describe the methods that you want them to use, and provide the limitations and provide the scope of the test guidelines. But then if you have an open, what will happen is that if you have something that is not as clear like that, I will suggest you put that either in the annex or either you do a guidance document for it, which is another option.
Yeah. Okay.
But Eric will save it here.
Yeah. Eric.
Thanks. And I'll try to save Mar here. I think it also depends a bit a bit on what type of method we're talking about. Clearly, for particle size distribution, it's a very clear and unique property that so the FISCAM is probably more restricted to certain methods than having a test guideline on some toxicity endpoint, where you basically, in many cases, you see something like make sure that you are able to measure your test component, formulations like that, not
Yeah.
necessarily describing a specific method, and there of course it depends a bit on what the purpose is of a certain test guideline.
Yeah.
And how restrictive that would be, but I would go more for description of what is needed, so what should it fulfill, and then prescribing a certain method, because usually the method developments, as we've seen with the SPI-ICP-MS, is much faster than and so to make sure that you have a future-proof test guideline, don't go into too much detail there, if possible.
Yeah, good, thank you. Next slide, Nicolina. So this is really a free text bit for anything that we haven't captured so far. You basically
yeah, exactly. And yeah, and just to press submit once you're done with your comment, or even a haiku if your heart's desire.
Yeah, good. And I will leave this open for a little bit, but I am well aware that we are half an hour of inspired time over our initial plan. So please put down here anything that you wish us to pick up as we go through the next parts of NanoHarmony. Anything we didn't cover today that is relevant for how we make 2 and 3 that more effective. Things that you stumbled on that didn't work, that we haven't answered today in terms of how you make them as smooth as they can be. I'd like to thank Thomas and Ma and Frank, who's already had to leave, for the inputs today. I found it was really good. It's amazing how long I can spend in NanoHarmony and learn something every time people talk. It is something where people build up, as Ma says, big corporate memory, but it's really hard for people to access that when they're stood there wanting to propose their test guidelines. So that's what we will work between NanoHarmony and NanoMet in terms of writing that corporate memory down as far as we can, and write the guidance that we can have a legacy from NanoHarmony in terms of that, and hopefully through NanoMet, Mark can adopt it into the OECD and have it sat there. Is there anything either of you wish to say at the end, Thomas or Ma?
Certainly, I'm very happy, first of all, that we had this discussion and this possibility of presentation, and what you, Klaus, most certainly will say as well. Any comments, please send to us of NanoHarmony, and we have to think of different levels, how we can make use of this types of the information we have gathered here in this session. And that's why I put in something like the training material. It's good that we have this webinar, but if a new person, scientist is coming, how can we make this person aware, and how can we help? So something like these ideas, and what can we do with it to improve in the future? I think that is important.
Yeah, excellent. Thank you.
No, nothing for me. I was just reading the suggestions there, and I think yes, I mean the communication. I think the communication has improved since NanoHarmony was launched. To be honest, because it's something that I think we are not very aware about the interest that others can have in our work, because sometimes it's so specific and so close, and something that I have learned with NanoHarmony. Yes, I think the use of the academic research and translate that into a regulatory purpose is some of the key issues that we need to continue advertising, and something that we can continue between NanoMet and NanoHarmony to explore how to improve this process. So far, to be honest, I mean, what I have seen is all the points that have been made are not nano exclusive.
No, exactly.
They're really test guidelines and guidance document, and those are the hurdles that anyone coming into this end will have to resist and try to keep going on. But I hope that also the OECD Secretariat can also help a little bit to mitigate some of those pains in the process. At the end, you have nice babies, you have nice test guidelines, and it's a big achievement.
Yeah, and I'd be very much with you there that it would be lovely if this wasn't just a nano set of guidance, but actually the guidance was as widely applicable as it could be in terms of standard setting. There's some interesting comments here in terms of the EU having a good use of their academic research, but I think that's because the academic research that's funded through the EU is quite, or has been at least, quite closely driven into needing to provide standards as well. Yeah. And that might affect how funding can be achieved in different lead countries, I guess. You know, if it is coming from a regulatory request, then there's possibly more likelihood of there being centrally national money to fund things like the ILCs. Whereas in the EU, we've kind of gotten ourselves stuck a little bit between, well, the EU funded the EU academic projects to develop it, and then we sort of had a catch-22 of how do we then standardize it in the next project, which we couldn't work out how to then source. Excellent. There's more coming here, as Thomas said. If you have anything after we close this, which I suggest we'll do in 5 minutes, but keep typing if you are typing, then send it to us by email and we'll stick it in manually afterwards. The last slide, I believe, is just, while we let this one run, an advertising for the phase 0, which we will run in early to mid-June. We're just going to have to move a few things around because other meetings moved, and find some availability of the intended speakers before we confirm that date. But that one, I think, is really important in terms of setting the scene for how, as Mara says, do we keep the maturity of the SPSFs as high as we can. The more issues you can address before you start, the easier this phase 2/3 bit should be. And with that, I would like to thank you all. And I'm sorry we ran over, but